thank you very much. I'm pleased to be here. As I was introduced, TumorGenesis is a wholly owned subsidiary of Predictive Oncology. It's a NASDAQ-traded company. The symbol is POAI. I'm also on the board of directors, as mentioned. Now, I'm going to be talking about two companies, and the reason we're talking about two companies, although the lead slide is about TumorGenesis, which I'm responsible for. Holomics is the second company that is owned by Predictive Oncology. And what the difference between the two is that Holomics is a fully certified CLIA certified laboratory that is able to handle and focuses mostly on oncology. And I'll talk a little bit more about that. TumorGenesis is focused really on the tools such as cancer cell culture technologies and other discovery tools that we're going to be introducing that actually Holomics would use in its client business. It will do clinical trial, basically, discovery technologies, be able to receive samples from clinical trials. And so we're trying to address this unmet medical need. We all know in the pharma industry and biotech that the genomics big data hasn't yielded that much in terms of success rates for targeted therapies. They are traditionally low, and the targeted therapies traditionally in oncology at least represent usually single-digit niches within a particular cancer. The uptake in clinical practice is even more sketchy. So there's a realization that, genomics just isn't enough. We really have to have more of a comprehensive multi-omic approach. And, that's the way to improve the success rate of our efforts in cancer, which are, as you can see from this slide, about as miserable as Alzheimer's. When I did have my own company, I owned a company called Pharmaco Laboratories, and we did a lot of work for companies, and in the Alzheimer's area in particular. So I had the distinct displeasure of watching my clients disappear and go bankrupt as their funds dried up and disappeared because their drugs failed in clinic. Now, really what we're trying to do is change the paradigm. This is the traditional model on the left-hand side. People can see that. It's one that everybody knows about. What we're really trying to do with rapid real-time cell, what we call living tissue banks, is to increase the number of screens of individuals and have them represented in the Petri dish. And, will this yield better success? Well, it obviously has to be better than what we're looking at over on this side of the screen, which is a really low hit rate. And really what this means is in the repurposing world, this was actually done on a metastatic breast cancer patient. And they evaluated a whole screen of drugs across the paradigm. And of course, the drug responses were shown to be effective. Only 3 out of the 189 FDA-approved drugs showed a reliable in vitro response. And then based on that, the oncologist changed the patient's therapy. So that is how it works. Now, this answering the need, TumorGenesis and Holomics will actually provide rapid access to actionable, what we call multi-omic data. And this is all done through the incorporation of artificial intelligence, machine learning, both from the cancer cell up from TumorGenesis, as well as from the patient sample, from the CLIA laboratory patient sample down back to the cell. So we'll meet somewhere in the middle. Now, this is really another slide that just kind of shows you I want to call the abysmal rate of being able to culture oncology tumor cells in the laboratory versus something like microbial, and people will say, look, this is 90% of the microbial bacteria that are in animals and humans. It's not 90% of bacteria that are out in the world, because there's quite a bit out there we haven't cultured yet or identified. Now, what this does translate into is, the HeLa line it's still used today, as are the NCI-60 cancer cell lines. These are immortalized cell lines, in the HeLa line. It's had its, I don't know what it would be, 69th anniversary. It doesn't represent what a tumor cell or a tumor mass looks like in a human. Now, it was essential for this early discovery purposes, but it's time to move on now. This is traditionally how this is done. This is, the tumor sample hits the path lab. It's cut up into small pieces. There's a liquid medium. You replenish it, once a week. You continue to do this until you've killed out all the other subtypes except the rapidly reproducing one. And then you study the genetics, make sure, and then you do the drug profile. And then finally, if you want to validate, the drug response, you can stick it into a mouse model, run it directly, or run it from, the drug response into the mouse as well. So you can do both. And then some people are also taking the tumor pieces and sticking them directly into the PDX mouse. Now, the problem with the PDX mouse is they're expensive. It takes a long time, and the tumors don't always grab in the mouse either. Now, what we're trying to do here is we're trying to change this paradigm of weeding out all of the other heterogeneous tumor types that are in a cancer cell. Now, we've been very successful with this particular media that was developed. It's called the TumorGenesis Inchi media, and the cell line. So the success rate that we've had is about 95%. We're able to take all subtypes. It's from the xenograft phenotype to genotype, and then I'll show you a slide or two that shows it correlates very well to the patient. The good news in this particular standard line is that if you have, for example, a metastatic breast cancer immortalized cell line, and the drugs that respond to that are very sensitive, in all likelihood you have a better survival if you have that particular marker. In our case, we're seeing a variance across the different subtypes, and we're focused right now on ovarian cancer. So we have 25 different cell line subtypes that show varying response to different oncology drugs. And that, I can tell you, is quite troubling, because that means you're not actually treating the tumor, you're only treating one cancer cell in the tumor. Now, what we also have seen is that histologically, this is the tumor sample, and then this is the grown in a dish. So it develops the structure, histological stains the same. And as you can see across the cell line and the tumor, there is a continuity across what we would call the DNA, RNA, and proteomic signatures. Now, what this really means is that this is what a histologist looks at. And so we're trying to get the histologist's attention and saying, look, if you harvest the tissue from, the surgical oncologist will harvest it. They'll send it to the path lab. If you use our media to grow these in the ovarian cancer, you're going to be able to grow them up and histologically stain them, and they're going to look like this. They're also going to retain their proteomic signatures, their DNA, probably close to 95%. And there's already published models on this, so we have articles that are available. Now, how did this all develop? It started back in 2001, and actually in Bob Weinberg's lab at MIT's Whitehead Institute with Dr. Inchi. And then it has developed over the years, and we licensed this technology as we thought it was. And we're also adding some additional new technology that is forthcoming as well. So this is what there's a couple of manuscripts in publication. as we all know, this, manuscripts sometimes take forever because there's multiple people on them. But, as these come out, we'll be posting them and making them available. Now, really, let's talk a little bit about the market, and I'm going to get back to, believe it or not, artificial intelligence here, but I just want to set some of the stages. We've all seen the rapid growth in 3D cell culture, and the global market this is a 2018. It's probably, I don't know where it is now, probably 21, 22 billion. It's a sizable market. Now, heterogeneity of tumors is arguably relatively new. The NCI-60 cancer cell line has been virtually abandoned. It still can be used for primary research, but it has very little bearing on the survivability of an oncology drug in a clinical trial. And what they found out is that the heterogeneity of the tumor does cause a problem, but what we're doing is, as laboratory researchers are doing, is they're using what we would consider to be unvalidated media, and they're just using standard media and culturing up their cells. And what that does is it changes the DNA, RNA, it warps the cancer cells, and it creates this irreproducible preclinical experiments that cost the industry about $48 billion a year. So, driving 3D cell culture to what we call the living cancer cell bank is really where this is going, and we're linking it to artificial intelligence. And what our goal is to treat all the heterogeneous cancer cells in the tumor. That is the new paradigm. All right, so how did we get here? This is a media that has been extensively used across mostly not-for-profit. The Italian National Cancer Institute has used it. Harvard, UPenn, you can see the number there. So what we're doing now is we have a GMP production facility that can produce on demand in the United States large quantities of 8 different unique medias exclusively focused on ovarian cancer to start off with. We're branching out into leukemia, breast, and the rest of it will be announced as we go along. But with that, we also have what we call our discovery toolkits, and this is a biomarker discovery toolkit. So if you're a researcher and you're interested in ovarian cancer, you can isolate the 25 different types by, some of them grow really slow. 11 of them represent about 90%, 95% of ovarian tumors. You can grow those within those cell types, from your own cohort samples, for example, that may come out of an oncology surgical unit. And you can study them using these biomarker toolkits and study the, whatever you want to look at, whether you're also looking at discovery compounds as well. So in addition to that, we're porting on the artificial intelligence and the machine learning, because we're finding out that, there's an awful lot of proteomic DNA data that's there. And looking at that, you can't make heads or tails out of it. You need something far more sophisticated, an algorithm to be able to screen those compounds. And then in addition to that, we're going to be adding chemical library screening. Now, again, this is from TumorGenesis point of view. So our goals are, 60 different cancer cell lines. 15 of those already are, active tumors. The new cultures will be leukemia, sarcoma, breast, head and neck, and colon. So, this is more of the same, just what's in development, what's ready to launch, and where we're going with this. Now, I'm going to backtrack a little bit on Holomics, because Holomics was one of the first companies. They had a way in that slide that I showed you originally on how tumor cells were isolated, and you basically kill off all the other cells except the rapidly growing one. They had a program called ChemoFX, and ChemoFX was one of the first drug screening programs in a CLIA service laboratory. So They have quite an extensive list of 150,000 different tumor cases, 131 different tumor types. They have the drug response profiles to it. They have the biomarker mutational profiles. They have some of the clinical outcomes, and they're currently in the process of digitizing over 40,000 cases. So a histologist or a pathologist can look at a histology slide of these 40,000 cases, and people that are interested in drug development from the clinical side on down can use Holomics and this tool to help them further refine their drug development program. Now, this is a slide that shows you the number of patient samples that were collected, and they were collected over a period of time. So the sweet spot is about 120,000 cases, and there's also 10 years of survival data. And again, this is all cancer-driven artificial intelligence. So it's across not just ovarian cancer, which is our primary focus, TumorGenesis, as well as Holomics is ovarian at the moment. So there's a program that they call Pioneer. This is the PDX tumor platform. And then, of course, we have the AI-driven, we're also part of McGee's Women Health and the UK 100,000 project, which is a stellar project. We're one of the few companies, it's mostly institutions that are involved in this. As I said, we've got the biorepository, 40,000, this is all the paraffin blocks, but there's also 100,000 slides as well as tissues that are preserved. And then, of course, the network of 1,400 clinicians, 15 years, and we're doing now a 400 retrospective in ovarian patient tumor outcomes. This is a model everybody knows. I'm not telling anything new, but basically just to refresh what we're talking about. Again, Holomics is involved in the UPMC McGee study for ovarian cancer, also involved in the ovarian cancer and breast cancer in the UK. They have relationships for colon and lung, and they've just recently done some projects in thyroid cancer for a publicly traded company called Interpace Diagnostics that is interested in thyroid. And so how does it work? Okay, this is, it's basically surrounds, Carnegie Mellon's developed a program called QT Med Core, and this is an artificial intelligence program that actually runs you through this sequence, and you can do this about 40% faster, and it's been proven to show about a 60% cost reduction. QTMD, the company itself, has recently been acquired by Predictive Oncology. So Holomics is using this with the patient on down, and we're going to be using it from the tumor samples on up. And again, how does it work? There's a pre-training, you take all the data that you can collect. We collect it, from our own as well as from the public. You make some predictions, you do some verification. How do you do that verification? You do the predictions and then you do the actual studies and you feed back the loop. So this is how the artificial intelligence component works within what we see as the new paradigm in cancer oncology. What we aren't selling is we aren't selling data. We're not interested in that. What we are selling are actionable models, both TumorGenesis and Holomics. Holomics is arguably similar to, probably more advanced than Foundation Medicine was, have much deeper, much bigger library of oncology samples, as well as leads into 1,400 people who have evaluated the ChemoFX program. And so we already have a large amount of data that's available there. That'll make the AI machine learning predict outcomes. That's really what our goal is. I think that's the goal that everyone wants in oncology. When you go into, you know, Mass General here in Boston as an ovarian cancer patient, if you don't have the PD-L1 marker or ATK or one of those other markers, which represents a single digit of that population, you're going to get standard of care. Standard of care is Taxol, carboplatin, or cisplatin. 1 week IV Taxol, IP, and that's intraperitoneal after the debulking of cisplatin, and then alternating until you can't stand it anymore. That's what happens to a vast majority of the patients, and no one knows why only 40% of those women survive. Well, with this artificial intelligence and with isolating the ovarian tumor subtypes, we may have a chance at doing so. And researchers across the globe can order the media. We also work in grants. You know, we're happy to collaborate in grants. If you're a pharma company and you're interested in a bigger picture program, then you call Holomics. But you know, underneath it, we can also do what I call the wet work in the lab, and we have the ability to generate huge amounts of data from both ends of the spectrum here. It's what we consider to be a unique cancer cell culture techniques and tools for discovery research, which includes identification of new biomarkers. Those kits are going to be coming out. And then, you know, reliably, we have to pay attention to the heterogeneity of tumors. And in order to do that, you need to organize all of that information, and you need to be able to access it. And that requires artificial intelligence, machine learning, and actionable, reliable drug screening. Now with that, if you're interested, you can contact, you know, Mark Collins, or you can contact me. So if there's any questions, I'm happy to take them now. Thank you very much.
And Richard, there was a question from the CEO of Sage Medic, Chris Apfel, and I did text you as well. He's asking to learn more about your cell line and media. So I did send you his email address.
Oh, thank you very much. Yes, I'm happy to do that. Anyone, you can I'm pretty visible. I'm also on LinkedIn. You know, so you can email me directly. This is my cell phone. Call me. If you're interested in medias, we do have a GMP facility that manufactures it on demand. And we're going to be rolling that out probably within the next month or so. But, you know, this COVID-19 crisis has really dampened everything. So I don't know how else to say it, but, you know, entire labs just shut down, period. I mean, Harvard was shut down, I don't know for how long.
Yeah. Richard, we do have a few questions for you.
Oh, sure. Happy to answer.
The first one is from Laura Prendergast. Do you have entire genome of all of these different tumor cell lines, the 60, 131 sequenced already or part of sequence?
You know, I believe we do, because there has been quite a bit of work, and there's published papers that if you send me an email, I can be happy to send you those publications that show in depth the genetics as well as the proteomics that were done. Now there's a whole bunch of groups working on this that we're currently supplying media to. And so there's probably 3 or 4 publications coming out in the next month or so as well that go into real in-depth on finding new, even within the 25 types that we've isolated, there are subtypes within those. So this is a little bit like triple negative breast cancer. You know, they started off with, I think they had 4 variants. Now I think they're up to 11. But then within each of those, they're also now finding subtypes of subtypes. And these, and those subtypes respond differently to combination chemotherapy. So you got a real problem here. And we're hoping to address it. What we want to do is get all of this out on the table, because you've got to get the pathologist the tools to make the oncologist change their mind on therapy. So if you can't do that, you're never going to get a change in the treatment protocol.
Right. Okay, we have another question. This one
Sure.
is from Junyul Kim. And the question is, do you also put an effort to transfect foreign DNAs to your cancer cell lines?
No, we don't. No, you know, that's certainly something that we've got more than enough to do right now. But that is, it certainly, I'm sure there's somebody that's thinking about it, you know, doing all sorts of, I mean, just being able to isolate these 25 different ones. You know, I talked to a researcher in the UK, and he said, look, I've been trying to isolate stuff for 10 years out of the ascites fluid of ovarian cancer patients. And I got the media. Within 3 days, it was growing. And he said it was a eureka moment. I don't know how to say it. He said, I tried everything under the sun, nothing worked. And all of a sudden now I can grow them. So, you know, that's really what we're trying to do. We've got to be able to grow these different tumor cells. We've got to be able to store the data. We've got to be able to analyze it using artificial intelligence and machine learning in order to provide the data so that the pathologist recommendation in the hospital makes the oncologist change their mind about treatment protocol.
Seems like a little serendipity always helped our research efforts. I've got one more question for you.
Sure.
And this one is from Asfatilas. And his question is, what is the current biggest challenge to develop predictive models in oncology? Now that could take up a whole nother presentation. So
Oh my goodness.
I just want to give a top line answer.
Yeah, I will tell you this, you got to stop focusing on the latest craze. You know, you're going to have to go back and do the hard work of isolating these tumor cell lines. And I will guarantee you, almost guarantee you, that if you're a researcher working in Korea, your ovarian cancer patient tumor cells are going to be different than someone working in Brazil with the Brazilian population, almost guaranteed. So there is not only variation, heterogeneity amongst peoples, there's heterogeneity amongst people's tumors within those subpopulations.
Wow.
So this is a very complex problem. I really don't know if we're just gonna keep working on it. That's the short answer. And you just got to really do the hard work. You got to get your pick and go down in the mine and start hammering away and see if you can find a vein.
Okay, let's one more. It's a combination question, excuse me. It's from Jiraja Rahman. And the question is the media specific for only tumor cells? Can you use it for fibroblasts?
Well, you know, I don't really think so. Well, you probably could. I mean, you know, you'd have to try it. But obviously, when, you know, there's 8 different media types for the different ovarian, for the 25 different ovarian cancer cell lines. So there's variations within those medias. So if you know the DNA RNA signature of your particular target tumor, we would recommend a particular media for that. And you know, our goal is to be able to culture all the surrounding tissue as well. And you know, first of all, if you can grow them separately, then you can put them back together. Now think about that. You know, that's a wonderful tool to have, because the hard part is disaggregating the tumor. So if we can find a way to, you know, first of all, separate them, which is what, you know, the discovery tools that we're going to create, and then re-aggregate them again as discrete populations, including the normal tissue that surrounds the cells, you're going to be able to study a tumor outside of an in vivo environment, in an in vitro environment where it creates itself. It looks like a tumor. It has its companion cells with it, and that's what you should be screening drugs against. So the goal in the long term is to have that exactly. Yes.
Okay, Richard, thank you so much. That was an excellent presentation, and we wish you continued success in a very difficult field.
Well, we certainly,
jobs.
yeah, thank you very much. And we certainly appreciate this opportunity. And I want to thank Sam and the team and yourself for hosting this. And I'm looking forward to providing more information on this complicated field later on. So anytime I'm more than available. So just let me know.
Thank you, Richard. Thank you very much.
Thank you. Bye-bye. Bye.